Now, the most important part of any project is the hypothesis, or at least the question that you want to answer. So I'm using the legacy database here for the TCGA because I need to get some information quickly. So where is my RStudio? Here's my RStudio screen. So the next thing I need to do within my script is I need to define the two groups that I'm going to compare. Now in this particular case, I'm interested in a question relating gene expression within cancer. So what I'm going to do is pick two groups, and I'm going to look at the gene expression in the first group and the gene expression in the second group, see which genes are differentially expressed between the two groups, and therefore use this as an explanation as to why those two groups are different. It's really important that you think carefully about what your hypothesis is. This and most of the examples I'm going to do are on gene expression analysis. But you can do something like copy number variation analysis, or you could do gene mutation analysis. I'm most interested in gene expression analysis because this, at least partially, relates to how the cells change their function as the cancer progresses. Mutations largely remain the same or remain as a sort of background and accumulate some more mutations. But how those mutations impact the function of the cell can be quite difficult to unpick. So for me, it's easier to start with something simple, such as which genes are being switched on and off between two different groups. So to define the different groups, I need a list of their IDs. So the IDs within the Cancer Genome Atlas look like this. So TCGA-BH-A18G. They have this sort of general format. So what I want is a list of participants, members that are going to be in the first group, and lists that are going to be in the second group. So how do I go around creating those particular groups? So let's go to the TCGA. Now, as I said, I'm going to use the legacy one just because it's quicker to show you what's going on, but it will disappear in the near future, so I'm going to create another version with the more advanced tool. I've had a practice with it. It allows other sort of questions and options to be done, but I do think it makes the manipulation of the files and finding the names and IDs much more difficult. So I've decided I'm going to do a hypothesis related to breast cancer. And my hypothesis is going to be related to there's a difference in the gene expression between stage 1 breast cancer, which is when it's small localized breast cancer, and stage 4 where it's metastasis. Because what you're going to need to do is switch on the different genes which allow the process of metastasis to happen. So you're going to need to switch on genes which allow the cancer to break out of its local cellular environment, to break free from the local cellular matrix. So it's going to have extracellular processes you would expect to be quite common in changing, and you might also see some other processes related to motility and some relating to sensing, because it needs to know where it is when it's traveling around the body and it's metastasizing. So I've clicked on breast. So it's telling me there are 4,046 cases that are within breast. They are within two major programs, one which is FM and the other is TCGA, and there are some more smaller programs as well. Now, if I click on FM, well, I can look at this part of the window. So here I have the case IDs. So these are the things that I'm looking for my list, for my workflow. tells me which project it's in, primary site, what the gender the person is, how many files there are available. Now this next set of columns of data are quite important. So the first one tells me how many times the sequence reads of that patient have been recorded, so 8 times. The next thing, it tells me how much expression data has been recorded, 4 times. Then it's simple nucleotide variation, so this is the mutations, 16 files contain that. There are 11 files of copy number variation, 3 of DNA methylation, and there's 11 clinical files as well as biospecimen. And if I go over here, I can actually see the slide images of the sample that was collected. Now if I click on the FM project, you can see that the FM project's only recorded data on single nucleotide variation. This is not what I'm interested in. If there isn't a file in the exp column, which is gene expression analysis, then I can't use that data. So I can't use any of the data from the FM project because it doesn't record what I'm actually wanting to do my project on. I need expression data. When I click on TCGA, it is all there, so that's good. Now there are actually data from breast cancer and I don't know what this other project is, but I want to focus on the breast cancer project. Now there are different disease types, but I'm not going to focus on those ones, and there are different sample types. So there's blood derived normal, primary tumour, solid tissue, metastasis and things like this. Now this has highlighted all 1097 cases, but I wish to divide that into 2 different groups based on whether they're in type 1, stage 1, or stage 4. So to do that, I need to look at the clinical tab. I need to go down from the clinical tab and I need to go to find the AJCC pathological stages. Here you can see how many cases fall into each of the different stages. So first group, I want to have stage 1. Now stage 1 also has stage 1A and stage 1B. So if I select all of those, I have 183 cases. That's quite a good number. Now it is displaying 20 a time on the page, which is a little bit inconvenient for me to output that data. To output 180, I'd have 10 pages. So if I change it to displaying 100 cases per page, it's only across 2 pages. So what I can do is I've got all of these cases here. What I can do is I can export the current table of data as a TSV. So that's saved it as explore case table, whatever it is, 2004 to whatever. So that is the first 100 cases. I go to page 2 and I press the button again and I export the TSV again. So now I have all of these case IDs, which are the type 1s. So that's my first group of data. So now what I can do is I can go to download and I can open that set of data and I can manipulate it. Now what I want to do is manipulate it using Excel. I'm going to go down and I'm going to find Excel. And I'm going to go to Excel and I'm going to tell Excel to open a file. I'm going to browse downloads, and I'm going to go open. I need to specify all files, and I'm going to open the TSV file. It's a tab separated variable file, so it's delimited, so that's correct. Press next. It's delimited by tabs, so that's correct, so press next. and then press finish. So here is the first file which contains 100 cases. I now need to open the second one. Delimited, yes. Tab delimited, yes. Finish. So here's the second set of cases. What I can then do is highlight this 83 cases. Control C to copy it. Go to the end of the other list. Press Control V to paste it. So now I've got my 183 cases, and I can save this as an Excel file. Save as. Save it as an Excel file to make life easier for myself in the future, and then name it something sensible. So TCGA, and this is stage, it's breast cancer. Breast cancer stage 1. Save. So I did that so that I can get the IDs, and that is important.